Hello, everyone. My name is Hannah Jandrin, and I'm a sophomore biochemistry student at West Virginia University. And my research under Dr. Geldenhuys's lab focuses on the role of MitoNEET in mitochondrial function and how it assists in the protection against age-related oxidative stress and neurodegeneration. MitoNEET is a mitochondrial membrane protein and has been proven in previous studies to show increased protection against oxidative stress when in association with TZD class compounds. The structure of MitoNEET can be observed in Figure 1A, and in Figure 1B, you can see the structure of the novel TZD class compound that we will be looking at in this study, known as CI987. So our purpose for this study is to evaluate the role of MitoNEET in the aging brain, and we want to do this by looking at how the absence of MitoNEET affects oxidative stress, as well as how CI987 acts as a ligand for MitoNEET. In this study, we used a wild-type strain, and we developed a transgenic MitoNEET knockout strain of C. elegans, and we confirmed this knockout was successful through the Western blot shown in Figure 2. And Figure 3 shows the experimental timeline. So we first aged both worm strains to day 5 of adulthood, and then we induced oxidative stress in them via a 2-hour paraquat challenge. Then we treated the worms with varying doses of CI987, and after this, we performed a cell rock screen staining to evaluate oxidative stress in each treatment group. Here in Figure 4, we have the cell rock screen stains for both the wild-type worms in the left column and the MitoNEET knockout worms in the right column. In the top row are the basal oxidative stress, and the bottom row are the stains for after the 2-hour paraquat challenge. And in the bar graphs to the right are the graphical representations of these stains, and the left bar graph are the basal stains, and this data showed that there was a statistically greater amount of oxidative stress in the knockout strain than that of the wild type strain. And the right bar graph shows the 2-hour paraquat challenge strains, and there was no significant difference in oxidative stress between these two strains. Here in Figure 5, we have the cell rock screen stains in the wild type strain. Each group underwent a 2-hour paraquat challenge as well as respective CI987 treatments. And the fluorescence graph underneath of the stains shows that there was a statistically significant decrease in oxidative stress going from lower to higher doses of CI987. In Figure 6 are the stains for the MitoNEET knockout strains, which underwent the same dose response treatment that we just observed in the wild type worms in Figure 5. Underneath of the stains is the corrected total fluorescence bar graph of these stains, and we saw there was no statistically significant differences between the dose response groups of the knockout strain. So in conclusion, the loss of MitoNEET increases basal age-related oxidative stress, and we observed that in Figure 4 when we compared those basal oxidative stresses in our stainings. In addition to this, we saw that wild-type worms showed dose-dependent reduction in paraquat-induced oxidative stress, while there was no observed difference in the MitoNEET knockout aged populations. For our future directions in this study, we will want to include our mitochondrial consumption rates using our seahorse mitochondrial flux assay, and we will also want to look at basal and post-treatment ROS quantification so we can further understand CI987's mechanism of action. Thank you for viewing my poster and listening to my presentation. If you have any comments or questions, please leave them below in the comments. And if you would like to contact someone directly, please contact one of the emails at the bottom of this poster.